cell anemia is caused by a single mutation that causes a single amino acid change, a change in a protein letter from a glutamate to a valine, and this causes the protein hemoglobin to bunch up and form these chains and cause blood cells to sickle. And so, why does this happen? Well, valine is really valuable. It's an important amino acid. We actually call it essential because our bodies can't make it ourselves, but it's also really hydrophobic. This means that water doesn't want to hang out with it, and so normally it's found on the inside of proteins, kind of hidden away from the water. What happens in the case of sickle cell anemia is that a mutation causes there to be a valine on the surface of a protein, on the surface of this really crucial protein, hemoglobin. So hemoglobin is responsible for carrying oxygen throughout the bloodstream. And so your blood cells are chock full of this stuff. What happens in the case of sickle cell anemia is that now you have a valine on the surface of the protein and it can't hide in the interior of the protein, so instead it has to seek out refuge somewhere else. It has to seek out shelter from the water in another molecule, and it finds another a nice hydrophobic pocket on another hemoglobin molecule. This causes it to form these chains and form these like polymers, these long fibers that actually clog up the blood cells and can clog the blood vessels, leading to sickle cell crises where basically the blood vessels get all clogged off and blood can't get through, and they have these really painful episodes. So this is just one example of the power of how important a single amino acid's properties can be, and it's just one example of things that we can talk about in terms of valine. So let's talk about the amino acid valine.
So I'm going to start by talking about the connection to sickle cell anemia because I'm guessing that that's what most of you care most about. But then hopefully that'll hook you into more of the hardcore biochemistry afterwards. Things about how valine is a branched chain amino acid, what it's used for in the body, et cetera, et cetera. So let's start with that sickle cell stuff though, and that gives us a reason to talk about the hydrophobic effect and hydrophobicity. So valine is what we call a hydrophobic amino acid. So hydrophobic and hydrophilic, these are two different terms that we can use to describe whether water hangs out nicely with them. There's this thing called the hydrophobic effect, where basically water will exclude things that it doesn't like and force those things to kind of clump up and minimize their contact with the water. Much more on this in other posts, but basically what happens is that all of these molecules that we're talking about, whether it's water, whether it's individual amino acids, or whether it's a whole protein, they're made up of atoms, which are the individual units of elements such as hydrogen, oxygens, nitrogens, etc. Those are made up of smaller parts, subatomic particles. These include positively charged protons, which hang out with neutral neutrons in this dense central core called the nucleus, and then they're surrounded by these negatively charged particles called electrons. What's important to know is that in order to form bonds with one another, what happens is that these atoms kind of overlap their electron clouds. They share pairs of electrons. They can share one pair for a single bond or two pairs for a double bond. But when they're sharing, they're not necessarily sharing fairly. So some of the atoms are going to be what we call electronegative. This means that they hog the shared electrons. And if they're hogging those shared electrons, well, now they become partly negatively charged and they leave behind the things that they stole those electrons from are going to be partly positive. So oxygen is really electronegative, and so when it's bound to hydrogens to form water, the oxygen is going to steal those electrons, and therefore it's going to be partly negative. The hydrogens are going to be partly positive. If you've taken any physics, you'll know that positive and negative things attract one another, and therefore water is able to form these big interconnected networks. And they really like one another, and so we can think of water as being kind of sticky. Now, if you stick something into water, if water likes it, it'll let it into its network, and therefore we call those things hydrophilic or water loving, water loved. But if the water doesn't like those things, then those things are going to get excluded by the water. The water is going to kind of gang up around them, and this is going to force those things that the water doesn't like to clump together. And we call those things that are getting excluded by the water, hydrophobic. And so water phobic, fearing, but they're not really fearing the water just doesn't like them. And so the water is going to exclude them. And we call this the hydrophobic effect. This comes into play in a lot of places in biochemistry, but especially when we're talking about protein folding, because some of the amino acids are hydrophobic and some of them are hydrophilic. Now, if you think about what this means in terms of the consequences of a protein, if something is hydrophobic, it's going to be in the inside of a protein typically, and if something is hydrophilic, it's going to be on the outside of a protein. And this hydrophobic and hydrophilicness is going to help influence how the protein folds. And in fact, that's going to be the main driving force of a protein folding is to minimize the contact of those hydrophobic things with the water, get those hydrophilic things on the outside. So what makes something hydrophobic or hydrophilic? Well, let's think about what water likes. So we know water likes other water molecules because of that partial charge interactions. So if something else has that has a partial charge, water will let it in. If it has a full charge, water will let it in. But if you don't have any charge, well, then you're really boring to the water, and the water will exclude you and you'll be hydrophobic. When we're talking about hydrophobic molecules, we're typically talking about things that are non-polar. So polarity was that separation of charge. And if something is non-polar, it doesn't have that separation of charge. It doesn't have those charged and partial charge regions. And this is the case when we talk about just pure hydrocarbons, so hydrogen and carbon. And basically hydrogen and carbon, they share those electrons pretty fairly. And so they're going to be non-polar. They don't have that charge for the water to get attracted to. And if we look at valine, we can see that its side chain, its unique part, its R group is going to be just this like purple group. And so basically you just have these carbons and you just have these carbons and hydrogens, which are going to share fairly. So we classify valine as nonpolar, and we also classify it as hydrophobic because it's going to be, the water is not going to want to hang out with those. You can see that it's one of the most hydrophobic ones. The other ones that are more hydrophobic have even more hydrocarbon groups. But valine is classified as very hydrophobic, and so you would not expect to find it on the surface of a protein. Now, what happens if it is on the surface of a protein? Well, now you can get problems. And so this is the case that we'll talk about with sickle cell anemia. So sickle cell anemia is one example of a structural hemoglobinopathy. Whoosh. So basically hemoglobinopathy. So hemoglobin is this protein that transports oxygen throughout your bloodstream. And hemopathy, that's like a disease. This is affecting the structure of the protein. And so a structural hemoglobinopathy is when you have a mutation that causes the protein hemoglobin to have an altered structure that causes it to not be as functional. There are also diseases in hemoglobin caused by problems with hemoglobin that just affect the production of hemoglobin, and we call these thalassemias. But anyway, this hemoglobin, so with the case of sickle cell anemia, that's going to be a particular hemoglobinopathy. So it's going to be a specific mutation in hemoglobin that causes disease. There are other sickle cell diseases that have similar phenotypes, so basically similar problems with how this hemoglobin functions or doesn't function and causes problems. So this mutation in hemoglobin, we need to understand a little bit about how hemoglobin is made up of. So it's this protein complex that has these 4 subunits. It has 2 beta subunits and 2 alpha subunits, at least in the adult hemoglobin. And the problem with sickle cell anemia occurs in one of the beta globin chain. So basically it's able to carry oxygen throughout the bloodstream because of the heme groups. So these heme groups are cofactors, so they're not they're not like proteins, but they're helper molecules that the protein binds to, and they bind to iron, and then that iron is able to help coordinate or like hold on to oxygen. And what happens in the case of sickle cell anemia is there's a mutation in the gene for the beta globin subunit. And this mutation is going to swap out a glutamate, so this negatively charged amino acid, for a valine. So valine that we just talked about. And what this does is this is a dramatic change because we're going from one of the most hydrophilic, so one of the most water loved, to one of the most hydrophobic, one of the most water avoided. And what's going to happen is that this is going to cause, now you have that valine on the surface of a protein. We said valine doesn't like to be on the surface of a protein. And you can't just like refold the whole protein in order to move that valine inside, because then all the other things are going to be on the outside that weren't supposed to be on the outside. So it's not as simple as just kind of like hiding that valine inside of the protein. So instead, the valine is going to kind of seek out a hydrophobic spot to clump up with on another hemoglobin molecule. And this is going to cause these hemoglobins to form these long fibers. We call this like polymerization, when things are forming kind of like chains. And so these abnormal hemoglobins are going to polymerize into these long chains that are going to twist together in these long helical structures. These are going to cause the red blood cells to get misshapen and form these like sickle shapes. That's where the sickle cell anemia name comes from. And these can clog blood vessels, which is called vasoobstruction, and cause painful sickle cell crises, or basically very painful episodes where the blood flow is obstructed. This polymerization is favored in the low oxygen state, which I'll talk about in a second, but basically there you can have these low and high oxygen form, like shapes of hemoglobin. And because the blood is cycling from high to low oxygen conditions, as it's taking oxygen from like your lungs to your toes, to your lungs, to your toes, it's going from low oxygen conditions to high oxygen conditions, and it's changing between these different shapes. And the shape that is found in the low oxygen state is going to promote this polymerization. In this position, the valine is in a location where it can form an interaction with another hemoglobin molecule. But so that would be the taut state, whereas in the R state, this relaxed form that's found in the higher oxygen conditions, this is going to not form these fibers as much. And so because the valine, there's like the interactions aren't favorable in that condition, there's kind of like there's steric clashing. So basically things can't occupy the same space. And so if you were to try to make those interactions that you made before, well, now there's going to be stuff in the way. But this polymerization is going to be favored in this low oxygen state. And so as the blood is traveling throughout the body, you're going from high to low oxygen. You're going to polymerize and unpolymerize and polymerize and unpolymerize, and basically you're super duper stressing these blood cells out. So the blood cells this is happening, like you can go through the cycling like 4 times a minute or so, and so these blood cells are going to have a shortened lifetime in addition to causing this like vasoobstruction. And so yeah, so sickle cell anemia is a really big problem, as are related beta thalassemias and hemoglobinopathies. And so remember, hemoglobinopathy is when you have the structural hemoglobinopathy is when you have a problem that causes a change in the proteins like shape and dysfunction, and beta globin thalassemia is when you just have reduced expression, reduced production. And there are a variety of different mutations that can cause these. And typically these are going to be what we call a homozygous or recessive mutations. So basically you need 2 copies of it in order to two like faulty versions of the gene in order to have the disease. And so in the case of sickle cell anemia, you would have 2 of those faulty versions of the hemoglobin B, which this beta subunit, which is going to be, this is referred to as this HBS when you have the sickle cell mutation, the sickle cell anemia mutation. But then remember, there are other mutations. And so some people have like a combination of HBS and another mutation. If people just have 1 copy of the faulty version, that's typically enough to compensate. And this can actually provide some advantages in certain situations, which is like malaria, which is why that's why the sickle cell trait has persisted and is so common. But so the trait is when you just have like one version, one copy of it, but then if you have two copies of it, that's when you get problems. I should also mention that there's some really cool research going on into how to treat sickle cell anemia and related disorders. So basically, hemoglobin. I mentioned how there's like two alpha and two beta in the hemoglobin, adult hemoglobin. So this is HbA. Turns out in fetuses, they actually make a different subunit. Instead of the beta subunit, they make a gamma globulin. This is going to have a higher affinity of oxygen so that they're able to take oxygen out of the blood after the mom's already taken what the mom needs. Then the baby is able to have a higher affinity. So it's, even though there's less oxygen left, the baby is able to hold on to it. And so they make this fetal form of hemoglobin. This gets turned off when early after the baby's born, due to this BCL11 kind of putting a brake on it. And so there's various treatments for sickle cell anemia that are aimed at kind of like turning the brakes off with various drugs or with like gene editing techniques. And so that's something to keep an eye out on. But sickle cell anemia and related disorders are a big problem, and they deserve more attention and research. And yeah, but that's enough on that. But I do want to talk a little bit more about some of the terms that we use when we're discussing mutations. So in the case of sickle cell anemia in particular, we have this E6V mutation. This notation is basically, we can use notation like this, where here we're using the one-letter abbreviation. So glutamate's one-letter abbreviation is E. We'll get more into why later, but basically it's just they already used G for glycine, and they needed another letter, and so it was the next one, and it got E. Valine V, that makes a little more sense. So we put the letter that it was and the amino acid that it was in front of the position in the protein that it was, and then what it was changed to. So we call this E6V, saying we went from a glutamate to a valine. We can also basically say glu6-val. That would be another way to say it. Speaking of glutamate, sometimes you might see glutamate called glutamic acid. That's the same thing, basically. We typically use those terms interchangeably. Glutamic acid, technically it would have the hydrogen here, so it would be protonated. But in the body, this is going to be found in this deprotonated state, this glutamate state. But you might see it called glutamic acid, and don't get confused by that. That's basically referring to the same thing. When we're talking about the mutation is actually, in its strictest sense, the mutation is what happens to the DNA. So in order to make a protein, the instructions for making that protein are in the gene. So in this case, the instructions for making that beta globin were in a gene on chromosome 11. And that would be the DNA instructions. And then a copy of that is made, an mRNA copy, a messenger RNA copy of that is made. And that messenger RNA copy is then used to make a protein. And how the protein making works is this process called translation, where the ribosome, so this protein making complex, actually travels along that messenger RNA and reads out the instructions of the messenger RNA, piecing together the amino acids it specifies in order to make a protein. And how those amino acids are specified are by 3-letter codons. So these like 3 RNA letters are spelling one protein letter, and a transfer RNA, a tRNA molecule with an anticodon, so with the complementary codon, so the complementary sequence, so basically kind of like the opposite of it, is going to come and bind and carry the corresponding amino acid, and the ribosome will link those up into the chain. So different codons are going to like spell different amino acids, and in the case of valine, it's spelled by anything that starts with the GU. What happens in the case of with glutamate, you see that it's GAG and GAA. And in the case of this mutation in the hemoglobin, in this beta globin subunit for sickle cell anemia, you go from a GAG, so a glutamate, to a GUG, a valine, and therefore you're going from this very hydrophilic amino acid to this very hydrophobic amino acid, and then we get the problems. But the mutation itself was happening in the DNA, but it was causing a change in the protein. And so we call that, this type of change that it causes is basically a substitution. So you're actually changing one amino acid for another amino acid. Sometimes you can have mutations that don't change the amino acid because, as you see, if we look back at that codon table, there was like redundancy. So we had multiple codons that could spell valine, or multiple that could spell glutamate. And so basically, if you were to change it from one thing that spelled valine to another thing that spelled valine, you wouldn't get a change. So we would call this like a synonymous mutation, but this would be a non-synonymous mutation in this case because we're actually changing the codon to something that spells something else. And in this case, it's a missense mutation because we're changing it to a different amino acid. And it's a point mutation because we're just changing one specific position. Okay, so that was a little bit more, that was a little off track, but I think it's just important to know the terminology around some of these mutations, because although we often refer to the mutation in the protein, the mutation is technically in the DNA. Some people get very like pedantic about it and be like, no, it's in the DNA. But but we often just talk about mutations in proteins, but the mutations are in the DNA. They may or may not cause changes to the protein. And in this case, it does cause a change in the protein. And another cool thing about this change is because you're going from something that's charged, negatively charged, to something that is not charged, it's going to influence how that protein is going to behave, how it's going to move in a charged field. And so we have techniques in biochemistry that use electrophoresis. So basically use electricity to move molecules. We're often thinking, when we're talking about electrophoresis in the lab these days, we're thinking of some sort of gel electrophoresis, something like an SDS page or an agarose page or an agarose gel, where we're using electricity to move molecules through a gel. In the earlier days, they used like fancy fancier equipment, more difficult looking to work with, definitely. But basically, this is called a Tessellius apparatus, and it is going to move things from a positive charge to a negative charge. And the molecules, depending on how charged they are, they'll go in different directions, and then you can read that out. And so Linus Pauling actually used this technique in order to characterize how normal hemoglobin and sickle cell hemoglobin differed. And because you have that difference in the charge in the case of because of this mutation, you're going to be less negatively charged, more positively charged. Therefore, the normal version is more negatively charged, so it's going to move further towards the positive charge. And so he was able to see this when he looked at the results. He could see that some of it in the normal, you're more closer to the positive. In sickle cell, you're closer to the negative. And in the case of where you have the trait, remember that's where you have one copy normal, one copy abnormal. Then you saw like a split where you saw some of both populations. So really cool stuff. The charge was then further used in order to kind of scale things. So that was looking at the whole protein, but then you could also look at just protein pieces to try to figure out what was the actual change that happened. So use this like peptide fingerprinting, which is pretty cool. You like chop the protein up into pieces, and then you do separate those pieces by charge, and this time on this like paper, using this like chromatography with chromatography in order to separate these different molecules. And then Ingram was able to figure out that that's what the swap was. So more on this in another post, and it's a really cool story. The first like molecular disease where they were able to show that a single DNA letter swap could cause a single protein letter swap and cause a disease. So yes, so that was the sickle cell anemia story. And now let's get back to some of just the basic biochemistry. So valine, as I mentioned, it's a branched chain amino acid. And so I'm guessing that you can tell why we call it that. It looks kind of like it has branching in its side chain. The other branched chain amino acids include isoleucine and leucine, so you can see that they also have these branches. We'll look at these in the coming days. Leucine, basically, you have one carbon further out from the valine, and then the isoleucine is kind of like shifting where the branch point is. All these branch chains amino acids, as we'll talk about more in the, I think in my isoleucine post, I'll talk more about this. They go through the same branch chain amino acid breakdown process, involving this branch chain alpha ketone acid dehydrogenase complex. Basically, in the case of valine, what you do is you end up first you get alpha keto isovaleric acid, you transfer that nitrogen off onto alpha ketoglutarate, make that glutamate, and therefore this gets rid of that, that nitrogen group. Now you have a keto acid, which then goes through that complex, gives you isobutyryl-CoA, which goes through more steps, gives you succinyl-CoA, which goes through more steps and can give you glucose, or it can be used for other things. But one of the things it can be used for is to make glucose, because the succinyl-CoA can enter this tricarboxylic acid cycle. So this is sometimes called the Krebs cycle or the citric acid cycle, and it can be used to make glucose. And therefore, because valine can be used to make this, and this can be used to make glucose, we call valine glucogenic. So it can be used to make glucose or blood sugar. Also, like the other branch chain amino acids, valine is essential. So you're not going to find it on this chart, because we can't make it with our bodies can't make it. So if something is non-essential, that doesn't mean you don't need it. It just means that your body can make it from other things. In the case of valine, we can't make it from other things. And so we call it essential. We need to get it pre-made in our food. In terms of size, valine doesn't look that small. But what happens is, because it's it has that branch point really close to this like stem, it's going to have restricted motion. So even though it's small, it's kind of like it's got its bulk close to its backbone, and this is going to restrict its motion. So I talked a lot more about this when I was talking about glycine, how glycine with its hydrogen, it was really flexible, and how some of the bigger things we're going to see are going to be less flexible. And so valine is one that's less flexible, even though it's not really that big. And the reason why is because it has that bulk close to its backbone, and that's going to limit the rotation around this backbone. And the backbone is its rotation is already limited. What happens is that as those amino acids link up, they do it through these peptide bonds. Remember how I said that atoms could share one pair of electrons for a single bond and two pairs for a double bond? They can also sometimes do this thing called resonance stabilization or conjugation, where basically more than two atoms are sharing electrons, and they're sharing in a way that they don't have enough for like full double bonds for all of them, but they have more than single bonds for all of them, so they have partial double bond character. One reason why this matters is that it's double bonds, you can't rotate around them. So you can rotate freely around a single bond, but you can't rotate around a double bond. And so when you have these amide bonds, these peptide bonds, with these they have this partial double bond character, so you can't rotate around them, and you end up kind of having a chain of planes where there's only specific locations that you can rotate around. And how far you can rotate at those positions is going to depend on how bulky those side chains are, and especially how close to the backbone that bulk is. So in the case of valine, we have that bulk really close to the backbone, and so it's not going to be able to rotate very much. And so therefore, you're not going to find it in structures like alpha helices typically. And so unlike alanine, which we looked at, which was really was the most likely to form these alpha helices, when we talk about valine, it's not likely to be there. So the bulk close to its backbone makes it so that it can't adopt really tight angles and things like this. And so we're not going to find it there. We're also not going to find it on the outside of the proteins typically, but that would happen. That does happen in the case of sickle cell anemia, as we saw, and that can cause problems. Finally, we'll end with just a little bit about its discovery, as always with these posts, because I think it's kind of fun. Okay, so going back to 1856, a chemist named von Basenes, sorry, I can't pronounce these people's names. So he was trying to see what was in glands, and so he minced up pancreas and he extracted the things out of pancreas. He was looking and testing for a couple of amino acids, so we found some of the amino acids were known at this point, but not valine. He found leucine and tyrosine, and he found something that was like that acted a lot like leucine, but it didn't act exactly the same as leucine, and he wasn't exactly sure what it was. He figured out the empirical formula, so basically how many carbons per hydrogen, per nitrogen, per oxygen, whatever. And so he knew how many there were per molecule and compared to one another, but he didn't know how they were connected. And then so later in the 1880s, a couple of other groups found it in albumin and sprouts of Lupinus luteus and Vicia sativa. But they didn't try to figure out the linkages. And other scientists tried to figure out those linkages, but there were so many different atoms they couldn't figure out how things were connected. And so most of the original research on valine was just trying to figure out how they were connected. But they were still having problems because they were trying to make these things in the lab. And when you make these things in the lab, then you have issues with chirality. Talked more about chirality yesterday, a lot with alanine, how like you have like these right and left-handed versions. And if you make things in the lab, you get these mix of versions, whereas in the body, you only have a single version. So they were having a hard time trying to figure out the stereochemistry, so the way things were oriented in space. But then techniques got better. Fisher was able to then separate those stereoisomers, separate those different versions that they had made in the lab, nail down the correct linkage. And valine was therefore thenceforth referred to alpha amino isovaleric acid, where the valine, the name, comes from the valeric acid. And yeah, so that was Fisher did a lot of characterizing it, and including tasting it. We talked about how you should never taste things in the lab, but people have been tasting things in the lab, such as glycine, where they saw that was sweet. In the case of valine, D-valine, so the version that our bodies don't use, is bittersweet, whereas L-valine is all the way sweet. And yeah, so that is the basics of valine biochemistry. So we have a branched chain amino acid, like the other branched chains amino acids. It is essential. Our bodies can't make it ourselves. It goes through a similar breakdown process involving the BCAA or this BCKD. Mutations in this, by the way, cause maple syrup urine disease, and we'll talk more about that, this MSUD disorder. In the later post, valine is hydrophobic, so it's typically found on the inside of proteins. If you stick it on the outside of a protein, that protein could have problems, and this is what we see with sickle cell anemia. Also, because it has that bulk close to its backbone, it's going to be unflexible, but it is very cool, and hope that helps you understand and appreciate the power of valine.